what is inside spirulina that actually helping our body? Now you actually know the basic of it, but you might need to a little bit deeper in our immune system, how it could actually support our immune system or boosting or even make it stronger. So right now we have this PhD in bioorganic chemistry in Oxford University, UK, and he is a committee member of Japan Health Food and Nutrition Food Association, JHNFA, and board member of CRN Japan Association of Dietary Supplement. 我们现在有英国牛津大学林肯学院有机化学院生物有机化学博士，还有 JHNFA 日本健康食物与营养食物学会委员，还有日本 CRN 饮食补充学会董事会成员，还有我们的we have PhD Kimia Bioorganic University Oxford UK, and he is ahli jawatan kuasa Persatuan Makanan Kesihatan dan Makanan Nutrisi Jepun JHNFA, dan juga ahli lembaga CRN Jepun Persatuan Suplemen Pemakanan. Ladies and gentlemen, right now, right here, we have the star of today, Dr. Yasumasa called out to share us on ELKEN, Spirulina, and Immunity. Welcome.
Hi, everyone. Can you hear me?
Yes, doctor.
All right. Selamat pagi. 大家早上好。乔白山。Along Sosti. Salam Sawat. That's the all English. Good morning, right? In your local languages. Yes? Can still hear? Okay. Shall I go on? Just wave, please. Okay. So, good ELKEN morning. Because of the difficult time, so I cannot go over to give the seminar, but still I can talk to you through the Zoom. And today, I'd like to talk about the immunity and how the ELKEN Spirulina works for your innate immunity. So let's start with the first slide. Okay, so immunity is a self-defense network or biological process which produces the immune response to protect our body by detecting and eliminating the harmful foreign substances, cells, and tissues. So those foreign substances are called pathogens, such as microorganisms, virus, carcinogens, and pollutants like dust and pollen. If this system does not work, you may become susceptible to infections or developing allergies. So the mechanism is very complicated, you can see, and there are two major subsystems, namely innate immunity and adaptive immunity. Innate immunity is a natural function. First, the skin, mucous membranes, and saliva and tears physically and chemically prevent the invasion of foreign substances and the pathogens such as pollen, disease-causing bacteria, and viruses. In response to the invasion of the foreign substances from the point onward into the body, the phagocytes such as neutrophils and the macrophages mainly recognize the foreign substances and the pathogens and eat them up. So this action is biologically called a phagocyte. It plays a role in preventing infection. Now, can you go back the slide? A little, the number 3, slide 3. I'm going to explain number 3. Can feedback? Can I go back? One more. Okay. 1, 2, 3. Next one. Okay. No, stop. So this action is biologically called a phagocyte, and it plays a role in preventing the infection. Phagocytes are distributed throughout the body and activated in a few minutes to a few hours when a foreign substance is invaded. So on the other hand, the natural killer cells patrol in the body and independently attack those cells initially infected by the pathogens and the cancer cells, and leading to the apoptosis, which is a cell death. Foreign enemies, microorganisms, or toxins having their own marks as a non-self, which means they're not existing in our bodies, are to be recognized and removed accordingly. So this activity varies from person to person and the pathogens entering the body. So if you have active innate immunity, you may be able to eliminate or repel the pathogens by yourself. Not all the foreign substances can be eliminated by innate immunity. Adaptive immunity is a system that eliminates the foreign substances more efficiently when innate immunity cannot handle them. So it takes several days for adaptive immunity to be activated. And first, dendritic cells recognize the information on the foreign substances, pathogens, which are eaten and degraded by phagocyte cells like macrophages, and then transmit the information to lymphocytes, namely T cells, B cells, and natural killer cells in order to start the next attack. So while NK cells, natural killer cells, and macrophages, which are innate immunity, are further activated based on the information of pathogens. And then helper T cells. Can I go back? Not yet. And the helper T cells command the B cell to produce the antibodies in response to this information, and the pathogens are repelled. In addition, the helper T cells issue commands to killer T cells to attack cells infected by the pathogens. And the T cells and the B cells, having encountered pathogens that became antigens, memorize the information of antigens by accumulating in the lymph node. Then they become mute memory. Is that okay? And even if the same pathogen invades the next time, those immune cells proliferate quickly, and it makes them less likely to get sick. So in general, the conventional vaccination use this mechanism. Influenza vaccine is made of the virus with weakened infectivity or just a part of the virus. So in case of the influenza, vaccine against a specific type of influenza virus expected to be epidemic can be selected and inoculated so that the B cells can produce the relevant antibodies in advance. So by doing so, that when the virus of the same type of the virus invade, it can be attacked immediately by the antibodies. So however, the influenza virus changes its structure by repeating the mutations. Because of its different characteristics, it is often the case that the antibodies specifically produced by the vaccines cannot cope with a new type of virus. So since each foreign enemy has different characteristics, each pathogen is specifically distinguished. Even if you have experienced influenza, you cannot respond to a new type of coronavirus. So on the other hand, adaptive immunity responds to and attacks any foreign enemy. However, it needs some period to get ready. So by the way, this SARS-CoV-2 vaccines of the Pfizer or Moderna developed are different from the conventional vaccines and based on a similar concept, but developed by a new technology of genetic engineering. So there is a theory of trained immunity. Innate immunity can be enhanced by the experience of acquiring the immune memories based on these infections. The high level of immunity can repel pathogens in the early stage of infection without relying on the specific adaptive immunity. Immunity can be compared to defense forces of the country with ground troops, battle tanks, fighter jets, and elite missiles at the full use of advanced information technology. So this is very complicated system, and it's like a war in the modern society. Can I have next slide? Next slide? Okay. So in the process of innate immunity, macrophages recognize antigens together with dendritic cells. So phagocytosed antigens are degraded and information is transmitted to lymphocytes. When T cells recognize information, they transform to become helper T cells. And also, as a commander of the immune system, they secrete proteins, physiologically active substances called the cytokines, containing activators to surrounding cells. So they command the B cells to produce the antibodies. At this time, helper T cells differentiate into Th1 cell or Th2 cells by secreted the cytokines depending on the type of the antigens. So Th1 cell plays a regulatory role in a cell-mediated immunity that attacks pathogens or infected cells by activated macrophages and NK cells. Th2 cell gets engaged in the regulation of humoral immunity involved an antibody-based immune function. So these two types of the cell also work to suppress each other's actions and maintain the balance of acquired immunity. So it is believed that the higher activity of the Th1 cell is, the better immune status can be. Th1 cells react against enemies such as the bacteria and the viruses. They secrete cytokines such as okay. They secrete cytokines such as interleukin-2 and the interferon gamma, and issue commands to B cells to produce antibodies such as TGG, which directly attack the enemies. Since the information of the enemy is memorized by the immune cells, the antibodies are ready to be launched for an immediate attack if the same enemy invades again. In addition, the Th1 cells activate killer T cells and the natural killer cells to attack enemies and activate phagocytic cells such as macrophages to destroy the enemies. Th2 cells, on the other hand, respond to allergens such as mites, moles, and pollen. So they secrete a cytokine called IL-4, interleukin-4, and cause the base B cells to produce antibodies IgE, which counteract these allergens. Allergic symptoms occur when the immune balance is lost when with the excess Th2 cells. Next slide, please. So adaptive immunity is responsible for the attacks on the foreign substances that cannot be handled by innate immunity. So the roles are as follows. Even specificity. So even if you have had the mumps in the past, you cannot prevent measles. Even if you have got the flu, you cannot prevent the new coronavirus. Adaptive immunity does not respond to the different types of pathogens because it recognizes only the characteristics of the pathogens to immunize it. And the second, immune memory. The adaptive immunity remembers the previous infection. So like missiles, once you have it, you will not be infected again. Vaccines use this mechanism. So by inoculating the detoxified pathogens, the specific immunity can be acquired. And three, diversity. Although the adaptive immunity is obtained specifically, it tries to deal with any enemy to some extent. So it takes time, but it can attack against various foreign enemies. And number four, the self-tolerance. It does not attack our own body because it attacks against the foreign enemies different from the cells inherent in our body. Next slide. So we are born with the dominant functions of Th2 cells. Then, by experiencing the various environment and encountering the virus and the bacteria, the function of Th1 cells gradually develop and acquire immunity. Then the immunity balance is getting maintained. The reason why the allergic symptoms in a childhood disappear with the growth is because of the improved immune balance. So however, the people in the modern society have grown up in a hygienic environment and are less likely to be infected with a virus and the bacteria in general. In case that the Th1 cells are less stimulated under such a condition, interferon gamma production will decrease. On the other hand, in case of that Th2 cells are dominant and the number of people prone to allergic symptoms may increase. So but in the modern society, the people tend to have an imbalance in immunity. Next slide, please. So we have a biological defense system called immunity to protect ourselves from the microorganisms such as the bacteria and viruses. This system consists of the innate immunity and adaptive immunity, as I explained. So immunological memory is acquired immunity as well as the trained immunity based on the immune experience. So the innate immunity in the early stage of the infection is trained and enhanced by the experience of encountering the bacteria and the viruses. The foundation of innate immunity research is said to be based on the research of immunopotentiators derived from microorganisms from the end of 19th century to the middle of the 20th century. By the way, you may have the image of bacteria as dirty germ, but we live with the many kinds of bacteria, indigenous bacteria in our bodies. One of the typical examples is 100 to 1,000 trillion intestinal bacteria we have. Okay. So bacteria can be generally divided into the gram-positive and gram-negative bacteria in biology. So main difference is the difference in the constituents of the bacterial cell wall, which are distinguished by the identification method of biology, gram stain method. So gram-positive bacteria include beneficial bacteria as well as pathogenic bacteria. For example, a typical strain of beneficial bacteria is lactic acid bacteria. Pathogenic bacteria includes Staphylococcus aureus and Mycobacterium tuberculosis. Gram-positive bacteria have a peptidoglycan as a thick layer in the outermost shell of the cell wall that enclose the cells. On the other hand, gram-negative bacteria also include beneficial a beneficial one as well as pathogenic one. Beneficial bacteria include the acetic acid bacteria used for the acetic acid fermentation, Zymomonas bacteria used for the tequila fermentation, Xanthomonas bacteria that produce xanthan gas, which is a food thickening polysaccharide, and pentoi bacteria that coexist with edible plants and suppress the growth of the mold. On the other hand, pathogens include Vibrio chlorella, Salmonella, and Escherichia coli. So, as a matter of fact, according to the taxonomy, the Spirulina is a blue-green algae, but it's also called a cyanobacterium and is a member of gram-negative bacteria. Gram-negative bacteria also have a thin peptidoglycan membrane inside the outer membrane, as well as lipopolysaccharide, LPS. So the immunopotentiating effect of the peptidoglycan, the component of the bacterial cell wall, and lipopolysaccharide, LPS, sugar complex of the gram-negative bacteria have been known since the end of the 19th century and the middle of the 20th century, respectively. So those chemical structures are different for each bacterium with the various functions. Can I have a next slide? Okay, so the peptidoglycan is a component layer of the bacterial cell wall. It is a kind of the polysaccharide with a peptide with a 3 to 4 amino acid bound to the repeating structure of beta-1,4 bonding with the N-acetylglucosamine and L-acetylmuramic acid, and cross-linked to form a network. And the peptidoglycan is known as the component of the cell wall of bacteria, such as lactic acid bacteria in a dairy product, such as yogurt. In many cases, because of their common structure, the partial structures of the peptidoglycan are detected by the immune system and the biologically defense functions are activated. So it draws attention as a substance of potentiating the immune functions, such as actions on the immune response cells, including the macrophage, B cells, and T cells, enhancement and suppression of humoral immune response, enhancement of the cell-mediated immunity, increasing the secretion of the leukocyte, enhancement of the infection inducer, and the enhancement of the natural resistance. Excuse me. Gram-negative bacteria also have a layer of peptidoglycan as a component of the cell wall, as well as a lot of glycolipid called the lipopolysaccharide, LPS, attached to the outer membrane. Can I have the next slide? So recently, it comes to be known that the LPS has a macrophage activating effect. Immunity is activated by the cytokines produced by the macrophages recognizing the LPS. So in humans, macrophages are present in the muscles and bones. Presented in all the tissues in the body, such as lung, liver, intestinal tract, blood, brain, muscles, and bones. So the intestinal tract is tolerant to LPS. LPS is not recognized to cause unnecessary overreaction as a foreign substance, but rather thought that the production of cytokine is induced by recognizing the environmental information with LPS. Macrophages are thought to transmit information to adjacent cells in this way to maintain the homeostasis of the tissue. So a key to the innate immunity is a toll-like receptor, a sensor of the surface immune cells that recognize the unique molecules of invasion of the bacteria and the viruses. In the process of the evolution, human beings, higher organisms, have acquired a sensor called the receptor TLR by recognizing the characteristics of the microorganism by encountering the various bacteria and the viruses. So now I would like to focus on the bacterial-derived immune potentiating the complex substances. Can I have next slide? So peptidoglycan activate the immune cells by binding to the foreign substance receptor TLR2 on the cell membrane responsible for innate immunity, such as macrophages. On the other hand, LPS activate immunocomponent cells by binding to another receptor called TLR4. Peptidoglycan and LPS bind to the different TLRs. So this means that the synergetic effect on the macrophage activation is expected. So it indicates the possibility of obtaining the better immunoregulatory effect. So peptidoglycan and LPS are cell wall constituents of the gram-negative bacteria. So some members of gram-negative bacteria are the pathogens. In this case, innate immunity works when the characteristics of the cell wall are sensed by the receptors, followed by the actions of adaptive immunity. However, the presence of the peptidoglycans and the LPS does not necessarily mean they are pathogenic. So in case of the non-pathogenic bacteria, peptidoglycans and LPS are still detected by the receptors. So they would be misunderstood as if the pathogen entered the body or a tumor formed. So macrophages are activated, cytokines are secreted, and the immune memory cultivated so far is evoked, and the quantity and quality of antibodies increase, and innate immunity is activated finally. Can I have next? No, go back. So to evaluate the effect of the spirulina on the potentiating the innate immunity, the activities of phagocytic cells and the natural killer cells were measured after oral administration of spirulina to mice for 6 weeks. Sorry, can I have next slide? Yes. So this is an experimental research done with the ELKEN spirulina. So the ELKEN spirulina was given to the mouse orally to see the activation of innate immunity. So immune cells taken from the spleen were stimulated by the immune activation reagent concanavalin A. So then we confirmed how efficient the production of cytokines in the immune cells and the production of antibodies in the serum. So the results were, next slide. Okay, with the, under the manipulation of the infection has been changed, you can see the significant activation of macrophages in the abdominal cavity was observed. So 29%, or 57% up according to the increase of the dosage. And next slide, please. And also significant activation of a natural killer cells in the abdominal cavity was observed. So in the spleen, you can see the 57% up and also more than double, 114% up according to the dosage increase. So natural killer cell is much more active than without ELKEN Spirulina. Next slide, please. Okay, so after giving the immune stimulation, the amount of interleukin-2 production was increased significantly. So interleukin-2 is mainly involved in the T cell proliferation and the NK cell and the B cell activation. So no change was observed in IL-4. IL-4 is involved in the most proliferation of IgE and IgE, allergic reaction activation. So there's no allergic activation. And then no change was observed in IL-10. IL-10 suppressed the interferon gamma production in Th1 cells. So no suppression of interferon gamma production. And then after giving the immune stimulation, there was no difference in the secretion of tumor necrosis after necrosis factor, TNF-alpha. So TNF-alpha is involved in infection protection and anti-tumor action. Its overexpression sometimes leads to the development of disease such as rheumatoid arthritis and psoriasis. So the secretion of interferon gamma increased remarkably after the immune stimulation. So interferon gamma activates NK cells and the macrophages and plays an important role in controlling the viral infection and anti-tumor immunity. So you can see 32% up of interleukin-2 to activate the NK cells and antibody production, and 26% up for the interferon gamma, which activate the NK cells and the macrophages. Can I have next slide? And there was a significant increase in immune antibodies, IgG, IgM, and IgA in the serum, while IgE tend to decrease. So it's good to see that not all the immunoglobulin was increased, but IgE still stay the same or decrease. So no allergic symptom was observed, but IgG, IgM, IgA, those are increased around 20% up. So let me explain a little more about the immunoglobulin. Next slide, please. So IgG is the most abundant antibody in the blood, and it's important for the detoxifying the bacteria and the virus invading the body, as well as for the recognition of antigen antibody complex by leukocyte and the macrophages. IgM is mostly present in the blood. IgM is first produced by the B cells against the bacterial and the viral infections. And IgA is abundant in the serum, nasal discharge, saliva, breast milk, and intestinal tract. It works locally. IgA contained in the breast milk protects the digestive tract of the newborn babies from pathogens. And IgE is the smallest amount in the body, but it's greatly involved in allergies such as asthma or hay fevers. So can we go back to the next slide, the previous slide? Yes. So those IgG, IgM are involved in to fight against the detoxifying the bacterial virus are increased, and also IgA is increased. So that's very good for the innate immunity. Okay, can I have next slide? And next one again. No, previous. Go back. Okay. No. Forward. Go next. One more. Next. Stay. Okay. So our body, I'm going to explain about our intestines a little bit with the immunity. So our body has the complex and sophisticated immune functions. Among them, the intestinal tract has the largest immune system. So there are the immune organs called the Peyer's patch in the small intestine. So which is similar to the lymph node and the large number of immune component cells in the intestinal mucosa. And it's the place where this intestinal immunity is affected by the food ingredients. Immune cells, with their unique properties, have functions such as oral immunotolerance, suppression of the food allergy with suppressing the immune response to the protein antigens in the food. So protection against the infection in the intestinal mucosa and the control over intestinal symbiotic bacteria. And also the protection as the intestinal barrier. So there is the very important organs called Peyer's patch, which detect some antigens or some food ingredients, whether to activate the immune system or not, is depending on this intestinal functions. Next slide. So this is a diagram of how the intestinal tract has detect the virus, bacteria, and then recognized, and then they give those information of the virus, bacteria are transformed to help a T cell to make antibodies to fight against those foreign substances. So intestinal immune cells can also detect the information of the food, bacteria, and the pathogens, and send that information to the immune cells throughout the body to enhance the immune functions. So when the component of the spirulina reach the intestinal tract, the immune cells encounter the unique component of the spirulina. Next slide. So spirulina is known as the algae. So it is a very primitive organism, biologically classified as the cyanobacterium as well. So cyanobacteria are also classified as the gram-negative bacteria based on the structure. So its structure has a layer of peptidoglycan on a very thin cell wall and that encloses the cell, and the surface of the cell wall is covered with the lipopolysaccharide, LPS. So it is recognized by the many scientists that the peptidoglycan and the lipopolysaccharide, LPS, are bacterial complex sugars, as well as the various immunopotentiators with different mechanisms of action due to each chemical property. Peptidoglycan and LPS of the spirulina are recognized by the immune cells in the intestinal tract, and they would be misunderstood as the foreign enemies have invaded. And because of those structures similar to the one of the harmful bacteria, then the information of the substances is transmitted, followed by the command from the immune command tower to get ready for war. Next slide. So it leads to an increase in the activity of phagocytic cells and NK cells when there is actual invasion by the pathogens and the viruses causing the immune stimulation that train the immune functions efficiently and immediately secrete antibodies and strengthen cell-mediated immunity. So ELKEN Spirulina promotes the secretion of interferon gamma and works to enhance the immune response by Th1 cells, while the secretion of IL-4 is suppressed and the immune response by Th2 cells that causes allergies is suppressed. So in other words, it was found that ELKEN Spirulina is expected to have an innate immunity enhancing effect while maintaining the balance of immunity. Next slide. So as we have learned many times, how good the ELKEN Spirulina is, because the ELKEN Spirulina is cultivated under the highest quality control system and care about the any contaminant exists, and we check, and then the whole final product ELKEN Spirulina is processed under the highest quality of the state-of-art factory, so resulting the highest quality in the market. So ELKEN Spirulina can keep you under the good balanced nutrition stage, and then it also suppresses the reactive oxygen species to minimize the threat by reactive oxygen to oxidation. And it's also suppress the fat absorption, and it can optimize the liver function, and also the potentiating the innate immunity. So I would like to advise you to keep the ELKEN Spirulina always on your table for your health and the beauty and the physical optimization and anti-aging. So that's the end of my talk. Thank you for your attention. Terima kasih. 谢谢.
Thank you so much, Dr. Yasumasa. Such an amazing talk, and as well so articulate that right now we know how our immune system work, and as well how Dr. have actually found ELKEN Spirulina is so wonderful that doing a lot of amazing things in our body. So right now, after we know a lot about immune system and about ELKEN Spirulina, maybe you have some question in your head, right?